Yes. So I'm the R&D lab manager at Decellularization. So Decellularization is in fact the world-leading gelatin and collagen company. So you might figure out what I'm doing here in a supercritical CO2 conference. So we will learn out later. So Decellularization is a part of Dining Ingredients Group. So that's a US company with the headquarters in Texas. And they are focusing on production of pharmaceutical, food, feed, fuel, kind of all types of products with one big thing in mind, sustainability. So it's in a recycling economy. So Ruslo is part of Darling, and we are producing gelatin and collagen from slaughter waste, like pig skins or animal bones. So in fact, 2019, I didn't even know what supercritical CO2 was and didn't even know it was existing. Today, I learned even that I don't really know a lot about supercritical CO2. So thanks to talk with Faye. So it's really exciting field. And it's also exciting where we want to apply this one. So a bit more on the company. So Decellularization is a worldwide company. So we have plants worldwide, and we have headquarters in Zand, the Netherlands, and we have one R&D department which supports globally, which is based in Ghent. So this is the plant in Ghent. So it's in the harbor of Ghent, nice place, and we have close by the pilot plant. We have a production facility, of course, and in the expertise center here, we also have an application center. So the major objective of the R&D department is, of course, new product development, and like James said, it's always with sustainability in mind. So Decellularization has, in fact, three main segments where we work in. So we have the health and nutrition segment with our hydrolyzed collagen. So this is really nutraceuticals and a food supplement. We have the functional ingredients, so that's what you already know about gelatin probably, in the hydrobro or in the capsules from Capsugel. But this is a new field we also work in. It's a biomedical segment. So biomedical is really where we focus on four different markets. So we have one which is about tissue engineering. We have one about more complex wound dressings, advanced drug carriers, and also cell culturing. And today we will focus on the specialized tissue engineering, of course. So this brings us, in fact, to the start of the talk. So worldwide, there's a quite large amount on tissue organs. So there's, in fact, a shortage of donors, of donor tissues. And resulting in very long waiting lists. So I just looked up yesterday the numbers for the UK. So there are more patients waiting for organs than they have received. So there are also not a lot of donors. Of course, we don't want to die as well. So, okay. And of course, there's an extra problem with transplantation is that there's always a risk of immunogenicity. So that the organ that is transplanted can be rejected by the patient itself. So this two things has led, in fact, to an increase in the research where we have regenerative medicine of an effect that we do tissue engineering as a direct replacement for organs. So for tissue engineering, you would need, in fact, three things. You need the cells. Preferably, these are cells from the patient itself. We need a functional biomaterial, and we also need a technique to put them in a 3D structure. So that can be 3D bioprinting, but you also have already decellularized organs, which can then be, again, populated with cells from the patient itself. In here, we will focus on the functional biomaterials. And in fact, Decellularization is already producing gelatins and collagens, which are functional biomaterials. But maybe the most important functional material is, in fact, a biomaterial that mimics the natural environment of the cell that you want to put in. So what is this one? So this is, in fact, the extracellular matrix. So we can also abbreviate it as an ECM. So the ECM or extracellular matrix is, in fact, a complex network of the macromolecules that are surrounding the cell. So you have their structural proteins like collagen, laminin, fibronectin, but also proteoglycans, glucosaminoglycans, but also growth factors, hormones, cytokines, but they're all bound into the ECM. In vivo, so in living organisms, they have the major function is affect the mechanical support of those cells, so they link cells to each other. But there is also a communication between the cells, and it also is very important in the influence on the behavior of cells. So, for example, growth factors that are present in the ECM will tell a cell how they have to differentiate and which type of cell they have to differentiate. So that's why if we can make an ECM product, then we can have, in fact, a biomaterial that's mimicking the natural environment. So what you need to do for that is you have to take the ECM and you have to remove all the cells. That's why it's called a decellularized ECM biomaterial. So how do you do that? So I just give you an example. So you can have a brain tissue. So can be an allograft or a xenograft. So xenografts from animals, allos from another human, for example, not the same human. And you have to decellularize that one and you make there from that a brain the ECM. So how do you do that? So in fact, there are three big steps in normal. So you first have a cell lysis step. So in cell lysis step is where you do some mechanical or chemical treatments onto the tissue itself that really facilitates the breakdown of the cells itself. So you really open cells, and then you have more access to the internal of the cell. Mode of actions there, osmotic shock. So using distilled water or just an high ionic solvents with a lot of salt. Dehydration mainly makes cells loose from their ECM. We had also mechanical lysis, cutting, blending. So just in kitchen blender, for example, a homogenizer. It's that, it's still. Freeze-thaw cycles. So just, making ice crystals, break cells, and so on. We can also have saponification, for example, that really dissolves membranes, the cell membrane of itself. The second step is then the cell removal, and that's really cellularizing the materials from the cell itself. So the cell membrane, but also the internals of the cell. In normal circumstances, they use a lot of surfactants for that one, like Triton X100, SDS, so sodium dodecyl sulfate, CHAPS. You can also use solvents for that, so ethanol, for example, which also precipitates a lot of proteins. Acids and bases, they also use it, and enzymes. So they'll sometimes use proteases like trypsin or even nuclease. And then in the end, you still have a final pH. So you have to remove what's left from the cells. So mainly it's done by just washing in water or in PBS buffer, but also removing a lot of leftovers from the surfactants or from the cell removal step. In fact, in the end, you have a decellularized ECM tissue. Now, what is, or how can you quantify decellularized, being decellularized? The problem is, up to today, there is no real guidelines on this one, so no regulation at all. But it's generally adopted by certain people in the academic fields that you have to have no DNA, or the DNA content of double-stranded DNA content should be below 50 nanograms per milligram of dry tissue. The fragment length should be below 200 base pairs. And of course, they always do an histological staining. And this is an example of an hematoxylina eosan staining. And so the blue points that you see here are the cell nuclei, and the pink is the ECM or the cytoplasma. So this is not treated, this is treated, so you don't have the cell nuclei there anymore. This is an example of NAOFTA, by the way. Okay, so there's a problem with these protocols. First of all, it's quite harsh. They use detergents. We can have residuals of these detergents as well. So SDS is toxic. Triton X100 is on the REACH list of a molecule of high concern. So it has to be banned normally. Because it's harsh, you can have a lot of losses of ECM components. So you want to have as natural as possible. So you want to keep everything. But if you can't keep everything, yeah, you can have losses, which in fact compromises the biofunctionality of your biomaterial. And then, of course, you can also damage your ECM components. I said bases can denature, or ethanol solvents can denature proteins. So in fact, you can have problems with mechanical integrity. So in fact, you want to have the mechanical integrity of your ECM still present there. And then another thing is that quite very long protocols. So it's not exaggerated, but I said weeks. These protocols of washing detergents, so soaking it, can take up to weeks, especially for very thick tissues like cartilage. Other pre-treatments can have to be done 25 times because to have enough cell lysis and cell removal of your thing. And it's also not so easy to scale up. Okay, it's not a product that it's really produced in very large scale, but it's quite important to know. So that is why supercritical CO2 comes into play. And I call it this one supercritical CO2 assisted decellularization, because supercritical CO2 alone is not enough. So it's not super fancy. In fact, if you look at this scheme here, it just looks like a normal SFE, just normal extraction. But the big difference is that the tissue or the product of interest is in the extraction vessel, and what's coming out, what we extract, is in fact waste. So this can be fat, cells, DNA, whatever that you don't want. So everything was from the cell itself. It's a rather new field, especially the use of supercritical CO2 in this case. So it's the first example was from 2008, if I remember well. And with really some significant literature, and not that much, or only 6 to 7 publications per year. Since 2017 there have been a lot of progress in this field, but still there's a lot of things to be discovered. So maybe a small word, why do we use CO2? In fact, is the repetition of what Ray said this morning. Low viscosity, high permeability, of course, it helps to go into the ECM and take out the cells. Interesting for the CO2, especially, it can be supercritical at low temperature and relatively low pressure as well. Because if you go too high, you might be able to boil slow cook your tissue, so you don't want that. So and the pressure is also important, because if you go too high in pressure, you might also damage your ECM structure itself. Non-toxic solvent, of course, sustainable, maybe even more sustainable in the future. Chemical inert, it doesn't interact or doesn't react with the ECM itself. Non-flammable, of course. It leaves no residuals behind, it's just a gas, so it's completely different as detergents. And economical, but maybe not for this moment, but might be it was in the past more cheaper to buy CO2, but okay. And it's also quite easily scalable. Okay, so how do we think it works? We don't know. That's the big question. It's completely fully understood. There are two theories that might exist next to each other. And first of all is that it's just an SFE-like, so it's just an extraction. And in fact, what you extract are the cell membranes. So CO2 might dissolve into the center of these membranes. Maybe can also extract phospholipids, and that's why it just breaks open the cells. The cell interior can also be then extracted afterwards. So that's a first thing. What is sure is that supercritical CO2 alone is not enough. So if you see a phospholipid has a quite polar head and an apolar tail. So CO2 alone does not extract this one. So instead, unless you have a very high pressure, but we don't want that, of course. So therefore, we use some co-solvents to be added. But more also important is that we have some pre-treatments and post-treatments as well, after the CO2 and before the CO2 of the tissue itself. We come to that. We have some examples in the next slides on that one. And then a second theory is that there is an importance of the pressure and the depressurization rate. So pressure and depressurization rate pressure can burst and lose cells from the ECM. So that's what I told. If you do a depressurization rate too fast, you also have ice crystals, which can damage the cells, but it can also damage the ECM. Especially also on too high pressure can cause ECM structure to rupture, or can just deform your collagen or your other structural proteins in there. So these are things about the working mechanism. Some important parameters. This is just now a summary from what I found in literature. So the biggest important parameters are these are just a list. So you have the pressure, so it's not extremely high. So the highest I found was 350 bar. Temperature 45 degrees Celsius, not more, because otherwise you will just denature your ECM. You will just slowly cook indeed your tissue. The time of treatment, of course. So yes, and you can have a long run, or you can have multiple runs. Okay, so in the second point, or in the second option, you have, of course, the risk of having ice crystals formed when you depressurize too fast. The addition of co-solvent or other additives, this can be added directly into the extraction vessel. I have an example in the next slide. Or you can add them as a co-solvent with a co-solvent pump. Both of those are tuning the decelerating power of your CO2. I also found some difference in the type of process. So you have a batch process in this case. It means that you pressurize the vessel and you close it off. You don't have any flow anymore. You leave it there for 15 minutes, 30 minutes, and then you depressurize. A continuous process in this case means that you just have a flow of CO2 constantly. So CO2 comes in, CO2 comes out at a certain pressure and temperature, and C continues is just a combination of both. So you first have a batch, then it continues, maybe different things. What also was important was the depressurization rates and pre-treatments. This can result in disrupting or lysing cells, and also have a post-treatment, which is important for the washing, but I have some more examples. In the end, it always has to do with the balance. So you have to have the ECM structure as intact as possible, so meaning the structure, but also the composition of the ECM and the mechanical integrity. And in the end, you also want to have an efficient removal of what you don't want. So that's always a balance for these parameters to play with. Yes. So this is a typical setup. So this is in fact the machine that we have in our lab. This is an example with a co-solvent pump here. And another possibility is that you have co-solvents in your vessel itself. Then you have a kind of structure, and your tissue of interest is on top of it. It does not come into contact with the co-solvent directly. So these are two ways that you can work with co-solvents in the decellularization way. So in our case, we have both possibilities as well. So we have a big CO2 pump, and here in the back, we have a P50 co-solvent pump. So this is a P200, this is a P50. We have two extraction vessels, one big one if you want to scale up in the future. We never used it yet. And a small one just to do the tests. And we have only one collection vessel because we are not interested anyway in the waste products. Interesting here is that together with James, I think, and also with pore separation itself, we designed a basket. So this is normal basket, but with an insert of this basket with a variable sample volume. So in fact, these are just discs, and you can just change them in any order. So you can have in fact different sample volumes. And mainly use small the smallest one volume that we can have, like 20 ml in this case. And it also adds an offset, so you can in fact add some co-solvent in your system. And the co-solvent is not in touch with your tissue. So some words about the co-solvents. Ethanol is the most used one. I think in 90% of all articles we have read, it was ethanol. So it's of course tuning the polarity of your CO2. And then some of those articles that use 100% of ethanol, the others use 70% of ethanol, which is a big difference because that has 30% of water. So this is also, so yeah. So we experimented with both of them. So it's it just gives different results as well. And by the way, the tissue that we put in can be dried or can be moistured. So it can be just wet tissue or can be it also give a big difference in the results. Important is that the addition of the co-solvent, you have of course a mixture system. Then it can change a bit of the viscosity density of your CO2 as well. So you have to put that one into account. This is something I still need to learn more about this as well. And of course, if you use more polar additives, you also might risk to dissolve compounds of the ECM that you don't want to dissolve, like the glucosaminoGlycans. And if you extract, for example, these ones, you will also change the viscoelastic properties of your end product. So that's not really so we want to keep this one in as well. Other co-solvents that have been used is Digipon LS454, 3-butyl phosphate, limonene, peracetic acid. I also have seen that one, hydrogen peroxide as well. So they are not limited to only the ethanol, especially you see that the peracetic acid you will encounter in the later slide as well. Some pre and post treatments. So the pre-treatments is in fact, they induce cell lysis, of course, but also cell fragmentation, and just makes it, of course, easier to work with in the CO2. There is also a hybrid method in one of the next slides. So where they in fact use detergents and CO2. So first in pre-treatment with detergent, and then they put the sample in CO2 just to have an extra addition of this CO2 treatment. Pre-treatments can be physical. For example, cutting. This is blending or mixing it like soup. And this is a normal kitchen blender. So this is, I think this is liver tissue, by the way. This isn't a normal liver and just cut from porcine, not from. Chemical pre-treatments can be soaking in bases, also detergent, like in the hybrid method, osmotic shock. So all those things that really can help and are not and are still sustainable. That's what we like to use. And you also have enzymatic pre-treatments that have addition of trypsin proteases, but also nuclease or RNase post-treatments, pre-treatments. And in the end, sometimes you have a post-treatment, mainly washing or ethanol evaporation. So the ethanol is something that really can evaporate quite well. So that's what people mostly do. So let me give you some examples. And a first example is in fact the first paper that came out, which is about porcine aorta decellularized ECM. So we have here the HE staining. So before you have still the nuclei, after you have only the elastin present here. So in this paper, they already did quite a good job, to be honest. They already looked into the fundamentals of the subcutaneous CO2. So what you see here, for example, is that if you don't add ethanol to your system for decellularization, you can't even remove phospholipids. So what they'll do is they plot the residual phospholipids. So you really need a cool solvent, especially at the pressure that you are using. And in this case, it was 300 bar, I think. Yeah, here, 300 bar. So you really need co-solvents ethanol as a co-solvent to be able to extract your phospholipid. What you also tested is that, in fact, additional pressure is not, it helps a bit, but not that much. So it really has close to a certain plateau. And this is our batch process, is a continuous process. You see that you go to the plateau just faster. To just the kinetics of a continuous process is just faster. And this is an interesting one. So here they put the sample for 30 or 60 minutes at 300 bar. And then they release the pressure. And they did it all quite fast, 2 megapascal per minute, or very slow. And they saw that they can extract more phospholipids when you do it slowly than when you do it fast. So still, for me, I don't know how that comes. In the paper itself, they also didn't explain a lot about it. It was about mass transfer, or that you give your phospholipids the time here to migrate from the center of your tissue to the outside. So it's I think it was the rate 2 or the process 2 in the talk from Ray. The next example is the decellularization of fat tissue, so adipose tissue. So there they have some pre-treatments as well. So what they did is they washed it, first of all, to remove all the blood with water, and then they soaked it in ethanol. So this sample here was in fact really in touch with ethanol, and then they put the ethanol soaked material inside the CO2 vessel. You have a batch process, only 3 hours, so not flow, just 3 hours at 180 bar and 37 degrees. And they add also co-solvent ethanol in the vessel itself, so it's directly in the vessel. And what they then saw is that, in fact, your structural proteins were still intact. So they did some measurements, and the HAP, so the glucosaminoglycans, are more present than when you would have removed this one with hexagon or with SDS. So you have more glucosaminoglycans present in the end product, and you also have the growth factors. VEGF, for example, vascular growth factor, was still present in the ECM. They could all quantify those things. So it was in fact a functional biomaterial in this case. A third example, this is an ovine cornea, an aorta again. They did no pre-treatment, so it was a wet tissue. And the process itself, so they put 70% ethanol in the extraction vessel, and there was no direct contact with the tissue. It was in continuous flow in this case, so for 1 hour, quite high. And then they have they calculated as in 20 to 80%, so solvent to CO2 ratio. I think it was in volume to volume. I would like to work in rate to rate, but yeah, so they managed it volume to volume. There was also one post-treatment, especially for the cornea here. So it's after the CO2, it looked quite opaque. But you can rehydrate this one if you put it in glycerol. So you come from this one to a clear cornea again. So this is again a functional one. So important here to note is that the authors found that with the supercritical CO2 treatment, there was less shrinkage. So the ECM itself was in fact the same shape almost, maybe a little bit smaller. And also the mechanical properties were quite identical to the original tissue, not treated tissue. And with SDS you have more shrink, so you see this one is very thin and also opaque and much smaller than you had without the CO2. Of course, they also did the HA staining, so that's normal. This is a fourth example. It's cartilage and tendon. So these are first example with quite tough ECM material, so quite thick as well. There they had quite a big pre-treatment. So the pre-treatment, of course, you see that it's depending on the tissue itself. For example, in the cartilage, in this case, they really soaked it with a CO2 phthalic detergent, so the DVPOM. And then they did a process with supercritical CO2. So that's what in fact is in this case is that the CO2 acts as a washing step to remove again your hydrophilic CO2 philic detergent. And they did a rapid depressurization, but they didn't mention anything in the paper. But they showed it was quite fast. And then in the end, you still have a washing hour of 24 hours with PBS. So normally this takes about 2 days in total. If you would have done it with a normal pre-treatment, with a normal non-CO2 process, it would have taken up weeks. So this is cartridge is one of the most difficult ones because it's quite dense and quite big. A fifth example. It's again porcelain aorta, but in this case it was quite interesting. So they did some experiments with CO2 that was saturated with water. So and then they tested, in fact, how much your product, your ECM, was shrinking after the CO2 treatment. So dry CO2, that was quite a lot. So you have 50% of mass retained only. But if you have in a pre-saturated CO2, you still have water inside, so you don't really dehydrate your product. So that's why addition of water to the CO2 can be also advantage for the mechanical properties in the end. And other things in this paper that are really mentioned a lot of different co-solvents. And in fact, this paper went about the hybrid system. So they first had an SDS pre-treatment, and then afterwards they have in fact the CO2 treatment on the SDS pre-treated product. They also looked at the chemical properties of the aorta in this case. So native is here. So this is the hybrid, it's quite close to the native one. If you would have done it only chemically, so only with SDS, it's again much thinner and also not as strong. And with ethanol itself, it also it becomes stronger because in fact you dry out your aorta, and in fact you have a bit of cross-linking of the ethanol. Unfortunately, they never were able to remove the SDS completely. So even in the end, you have still some SDS present. So, and the hybrid system doesn't really complete. So in fact, what we would need is a system without without SDS even. And the last example, because the title was of animal and plants tissues. So I have one example of plants. So this is the spinach leaves. It's also DECM. It's a bit quite different than what you have normally for animal tissues. So you have a very harsh pretreatment. So you have washing with the hexane. In fact, that's to remove the cuticula. So the waxy layer on top. And you also wash it with PBS. And then you have different co-solvents that they tested. So, and one of the co-solvents here was peracetic acid. That was quite close, 2% of peracetic acid that they added as a co-solvent in a batch process. So it's again directly into the vessel itself. They also have depressurization rates quite slow, I must say. And then also a post-treatment with bleach and with deionized water. And in the end, you so this is a complete chemical pre-treatment. But this is what you could get with peracetic acids. It looks quite nicely. But most important here to show is that the treatment time is way smaller than when you have the normal chemical treatment. And the CO2 can already sterilize in the beginning. So if you want to have a sterilized product, you can in fact combine it. So this is just a summary now. So you have a native tissue, you have a pre-treatment, CO2 treatment, post-treatment. In the end, you want the product which is the optimal conservation, but without the DNA, without the cells, and without the things that you don't want. And this can then be used as a biomaterial for bioinks, but also for the tissue engineering itself. Maybe this is the last slide. So I talked about decellularization with effects. Supercritical CO2 has many more advantages as well. So other uses of CO2 are decellularization. So you can use CO2 as a sterilization agent, but mainly in combination as an alternative to gamma irradiation or ethylene oxide sterilization. This is an example. It's not that nice. An example, for example, for gamma irradiation. So this is collagen on an SGS page. So if you gamma irradiate that one, you in fact break down the collagen. So this is before and this is after gamma. So in fact, you really break down your biomaterial. So it's the sterilization of supercritical CO2 is quite soft sterilization and uses the addition, an additive, and mainly other oxidative additives. So you know maybe the Nova Sterilise equipment, but NovaKill is an additive, peracetic acid or hydrogen peroxide are just possibilities. There is no mechanical change, so there's no change in fact on your product, and it's approved in 2019 by the FDA as an alternative method for the ethylene and the gamma irradiation as well. You can also use CO2 for drying, mainly like in solvent exchange process that you have there. For example, if you have exchanging ethanol for CO2 and then you can remove it. Impregnation, what Rui said as well. So it's in fact, for example, adding antibiotics into into a medical device. In the end, can be a sponge, for example, here. So it's quite important. And also, I found it as a forming agent. So as a byproduct from if your, for example, your plastic or polymers melts, and your CO2 goes into that, and if you then depressurize, you in fact get the foaming. So it can also be something that you want. So it's mainly combined with PLA or polystyrene or something. So, okay. So with this, I would like to thank you for your attention. If you have questions.
Did the 100% ethanol or the 70% work better? Because I've done cell culture before and used 70% ethanol because it kills bacteria that you're trying to keep the area sterile by bursting viruses and bacterium out. Does it help break the cell wall in that way for this process, or does it make any better the results when you use 70% rather than 100%?
Well, I would.
I might have said it, but
I didn't say it, but it works better with 70%. So we did an experiment. It was not in the slides. I removed it. In fact, where you effect dry your complete product. So it was a dry level in this case, then, for example. And melts and granulated. And if you put 1% of ethanol, if you do an extraction with 100% of ethanol, it doesn't extract anything. I mean, from the DNA. But if you use 70% ethanol, it was almost free of ethanol. So you can do in effect two things. Or you have a wet tissue from the beginning and use 100% of ethanol, or you can have a dry tissue and use ethanol with water. I think that's and you in effect moisturize a little bit of your sample.
With the 30%, I'm guessing deeming.
Yes.
Could you lace that with other things like trypsin, etc., to enhance so essentially improve on your pre-treatment time by reducing it, having it in with the CO2, or that denature things enough that you won't work?
Well, trypsin is an enzyme. It's a protease. Might degrade your ECM as well. You can add that.
Yeah.
So we tested a lot of things in that one. We even tested sodium hydroxide, but that doesn't work. It blocks completely.
Yeah.
You touch sodium carbonates, and that's a solid, and it's just
eat. Yeah. And are there any issues when you rehydrate? So obviously using scaffolding, aren't you, effectively to grow back?
Yeah.
Something I think people want to see. Are there any other side issues you can post-use? Have you seen any issues with when it's been regrown or anything?
We are not up to that point yet, but based on literature, it should work better, to be honest, because it's more natural. Even you keep as much as possible.
Very interesting. So I've grown cells before the cell culture work with a robotic system, and I'm now doing supercritical CO2, and it's really interesting to see the crossover.
Imagine that's to scale up to this. It would be the size of a final unit. And second one, have you considered to combine ultrasounds with CO2 for the stabilizers, which is an efficient method for sterilization, but with high power. Just yeah.
So the size that we go to, I think it's not more than 5 liters vessels, because the product that you produce in that one, it's sold by milligrams, not by grams. Quite expensive as well. So yeah, if you have a 5 liter vessel, 1 kilo of tissue, dry tissue, then would be more than enough, I think. And the second question was sonication. I didn't test it together, but I just tested before as a pre-treatment. And the problem is with the pre-treatment, it just heats up your sample quite a lot.
Yeah, I was considering in high pressure.
In high pressure, we didn't test, no, but if you do it as pre-treatment, it was 30 seconds and it was up to 37 degrees from 20. So it was not even a lot.
I was going to say, if you blended it and you've got like a slurry, if you like, could you not make a continuous process potentially by feeding it with a scCO2 solvent pump type scenario, so that you could have like an in and then catch and then filter maybe. I suppose because you got waste in your outcome, it's harder to separate.
Well, we
couldn't continue it possibly.
Yeah, we are not interested in the waste in this case, but never thought about this one.
I was just thinking sonication. You can also use sonication that will blend the liver, etc.
Yeah.
I used to use a sonicator that would blend 3 cubic centimeters in about 30 seconds. Having an ultrasonic power in it.
Well, one of them, the waste is also a homogenizer, so a high pressure homogenizer that you go 2000 bar through a very small hole. It also bursts the cell, so it's also a good test. Yeah. We might be making a liver smoothie then. Again, there you have the problem with the pressure that can damage the ECM. It will also heat up your sample a lot.
Good.
Yeah. So the first experiment we did, we cooked pancreas. We really cooked it.
Cleanup must be fun on this one as well.
Yeah, well, James was lucky there still. Do you remember?
I do, yeah, I remember. And I'm like, oh. Fate is the way. Perfect. Right, then. Thank you. I don't think there's any more questions. Thank you very much. Thank you, Rebels.